When we deal with plant diseases, quite often we're dealing with a single organism that's causing a disease. But in the area of seedling diseases, there's multiple pathogens and there's multiple species of a particular genus. So you would have a huge complex in any given field that could be potentially involved in a seedling disease complex. We have some organisms that are oomycetes, and then we have some organisms that are true fungi, like the Fusarium and Rhizoctonia. Then in each one of those, we have multiple species. So that's where it's a challenge with regards to us doing control measures to manage them, but also to find out what in the world is going on in the field when we're dealing with problem situations. There's a United Soybean Board and a North Central Soybean Research Program grant that's been funded now for 2 years, and it's funded at the rate of about $500,000 a year, and it's addressing this critical question: is what is in the fields of producers in this region. So this covers farmers all the way from North Dakota all the way down to Arkansas, where they have universities that are involved in this research, and we're trying to get a handle on what is in the soil, what is most prevalent in this complex, and then we're also trying to develop tools that can rapidly diagnose which organisms are there. That's important for the person that's going to be making recommendations to the producer, but also for the companies that are developing germplasm for the producer or chemical control measures for the farmers, to know what the spectrum of organisms is there. And then when you have situations where maybe things that are being deployed to control the organisms, maybe they're not working as well, the information on what is actually there would tell us more about why something may be failing. The other thing that's a component of this project is to develop better ways to screen these organisms for just to determine how pathogenic or how much disease can they really cause. There's a lot of the organisms we're finding that are turning out to be beneficial organisms, whereas they may be antagonistic to the seedling disease pathogens. So we're trying to get a better handle on those. We're also trying to develop assays where we can study these organisms. There's some organisms that we're finding that we've not worked with before. So we have them in culture now at these various universities, and each university is working on the pathogens that are most prevalent in their region. So it's kind of everything is complementary. We're working kind of in the same focus.